Welcome Genetic Teacher Channel. Today, we are going to explain how to use electron microscopes. Firstly, let's define electron microscopes. Electron microscopes are scientific instruments that is used a beam of energetic electrons to examine objects on a very fine scale, to see the fine details of the interior structures of organic cells, such as nucleus and mitochondria. In addition, the magnification object of electron microscopes is 100,000x to 300,000x, and a photograph taken using electron microscopes called micrograph. So, electron microscopes uses electron beam to produce image of the object, and magnification is obtained by electromagnetic fields. There are two types of electron microscopes: transmission electron microscopy, that need thin section and negative stain, and scanning electron microscope, which coat the specimen with heavy metal. Firstly, transmission electron microscopy was the first type of electron microscope and focused beam of electrons instead of light. In addition, it is used to examine cells and cell structure at very high magnification and resolution. Because of the wavelength of electrons is much shorter than the wavelength of visible light, the magnification is the power of the microscope to enlarge the image of an object, while resolution is the power of the microscope to show detail clearly. So, to study the specimen with electron microscopes, a series of processing are required, such as fixation, dehydration, embedding, cutting and positive staining with heavy metals. Firstly, cut tissue in about 50 to 100 nanometer thick, using ultramicrotome. Then expose the specimen to fixative container as soon as possible. Then, dehydrate the specimen by replacing water with alcohol to remove all free water in the fixed tissue, as follow: 1. Distilled water, 2. 40% ethanol, for 5 minutes, 3. 70% ethanol, for 10 minutes, 4. 90% ethanol, for 10 minutes, and finally, 100% ethanol, in 3 times with 10 minutes, every time. After that, stain the sections using lead salts, then, mount section onto a small copper grid, and finally, place the grid into the vacuum specimen chamber, as viewed in the figure inside the slide. The components of electron microscopes are 3 components. The first component is electron optical column, which involves 4 parts. 1. Electron source, to produce electrons. 2. Magnetic lenses, to demagnify the beam. 3. Magnetic coils, to control and modify the beam. And 4. Apertures, to define the beam and prevent electron spray. In addition, the second contains vacuum systems, that including 2 parts: 1. Chamber, that hold vacuum pumps to produce vacuum, and 2. Valves, to control vacuum, and gauges to monitor vacuum. Moreover, the third component of electron microscopes is signal detection and display, that contains detectors, which collect the signal, and electron, which produce image from the signal. Be attention please, special techniques of thin section are needed to prepare specimens before observation. Furthermore, specimen must be held inside the vacuum chamber, because electrons do not travel very fast in air, due to charge particle nature. And finally, the image is not directly visualized by eye, it is formed as a photograph, or as an image on a monitor. No, electron micrograph. Now, let's explain transmission electron microscopy. Transmission electron microscopy was the first type of electron microscope and patterned exactly like light microscope. It uses a beam of electrons to focus on the specimen, to produce an image. Because of electrons have a shorter wavelength, that results greater resolution. In transmission electron microscopy, the electron beam is transmitted from electron gun, through an extremely thin sample, in about 50 to 200 nanometer. The first condenser lens determines the spot size, or the general diameter of the beam, when it hits the sample, and the stream is condensed by the first condenser lens, controlled by coarse probe knob. Therefore, this lens is used to form the beam and limit the amount of current beam. In addition, the second condenser lens determines the brightness, or intensity, of the beam on the sample, by increasing or decreasing the beam diameter that hits the sample, and the beam then passes through condenser aperture, which filters electrons that are not tightly bound in the beam from traveling to the sample, and finally, the interaction of the electrons traveling through the sample and analyzed in two-dimensional image. And now, let's face scanning electron microscope. Scanning electron microscope is an instrument that produces a largely magnified image by using electrons instead of light to form image. In this electron microscope, a beam of electrons is produced at the top of the microscope by electron gun. After that, the electron beam follows vertical path through the microscope, which is held within a vacuum. Then, the beam travels through electromagnetic field and lenses, which focus the beam down toward the sample, and, once the beam hits the sample, electrons and X-rays are ejected from the sample. After that, detectors collect these X-rays, backscattered electrons and secondary electrons, and convert them into a signal that is sent to a screen similar to a television screen to produce the final three-dimensional image, as presented in the next slide. In details, electron gun, placed on the top of the system and used to produce electron beam, and have V-shaped tungsten filament cathode which emits electrons upon heating. Therefore, when electrons are transmitted from the cathode, they pass through columnar aperture to the anode at high voltage with constant energy, which is efficient for focusing the specimen to produce an accurately defined image. It also has the two condenser lenses system, which works to focus the electron beam on the specimen by controlling the energy intensity and the columnar aperture of the electron gun, and, the first lens, has strong magnification, produces a smaller image of the specimen to the second condenser lens, directing the image to the objective lenses for higher magnification. Furthermore, image-producing system is made up of the objective lens, movable stage or holding the specimen, and projector lenses. The objective lens has a short focal length, and it produces an intermediate image from the condenser which transmitted to the projector lenses for magnification, and the projector lenses are two-type: intermediate lens, which allows great magnification of the image, and projector lens, which gives a generally greater magnification over the intermediate lens. Therefore, to produce efficient high-standard images, the objective and projector lenses need high power supplies with high stability for the highest standard resolution, as exhibited in the next slide. And finally, imaging recording system is made up of the fluorescent screen used to view and focus on the image. They also have digital camera that permanently records the captured images after viewing, as showed in the figure inside the slide. In abbreviate, as described above, a heated tungsten filament in the electron gun produces electrons that get focus on the specimen by the condenser lenses in the column tubes under vacuum condition. In addition, vacuum prevents the collision of electron with air to clear formed image. Furthermore, condenser lenses are used to focus the beam of electron on the specimen, and finally, the specimen scatters the electrons, focusing them on the condenser lenses, forming a large clear image. So, electrons are easily absorbed and scattered on solid elements. Therefore, it shows poor visualization in thick specimen. Therefore, very thin specimen are used for accurate and clear visualization. So, the major objective studies of scanning electron microscope are: 1. To study topography, that is the surface features of an object, or how it looks, and its texture. 2. To study morphology, which is the shape and size of particles making up the object. 3. To study chemistry, by studying the elements and compounds that the object is composed of and the relative amount of them, and 4. To study crystallography, which study how the atoms are arranged in the object. Let's make differential between transmission electron microscopy and scanning electron microscope. The main differences between transmission electron microscopy and scanning electron microscope are: transmission electron microscopy uses transmitted electrons through the specimen to create the image, meanwhile, scanning electron microscope creates an image by detecting reflected electrons. As a result, transmission electron microscopy offers valuable information on the inner structure of the sample, while scanning electron microscope provides information on the sample's surface and its composition. This slide presented how the electron beam transmitted through the specimen in both transmission electron microscopy and scanning electron microscope. This slide showed the surface image and interior organelles of cells taken by electron microscopes. Finally, from the above data, the electron microscope's applications are: 1. To visualize and study cell structures of bacteria, viruses and fungi; 2. To view bacterial flagella and plasmids; 3. To view shapes and sizes of microbial cell organelles; 4. To study differentiation between plant and animal cells; 5. To study nanoparticles; and finally, to identify damaged microparticles which further enable repair mechanisms of the particles. Thanks for your attention. Please support Genetic Teacher channel by liking and sharing our videos and subscribe it. If there are any question, please comment us.